And good morning, one and all. Now, I don't know if you watched or looked at any of the videos, but I already got my first one, and this is bound to do it again, of comments. Someone was talking about how they had severe depression and herpes, and Dr. Ascot cured them of it. And here's his website. And I encourage you to check this website out. And it does slightly freak me out that the Google verse is essentially they are transcribing, doing a speech to text of what we say, or what I say, and that makes it searchable, because that's how people are finding this thing out. So it was last Friday that I was talking about anthrax and made some kind of offhand, I thought I'm using comment, or maybe it was about Coffee Mate in the Decatur post office cleared the building out. And I'm like, oh shit, I really shouldn't say that. And I thought about editing it, and then I posted it and went, oh, what the hell is it? So I'm waiting for the FBI. But they wiretapped me anyway, so whatever. I know. All right, so we're going to shift our attention to no longer prokaryotes, but to certainly a critical disease-causing agent, and that is viruses. And the viruses, particularly, or the term virus is you may have heard about like the computer viruses. It is particularly appropriate when we talk about computer viruses, in that you're talking about a little bit of programming. And essentially that's what viruses are, is they are a small amount of nucleic acid programming. I mean, that's what our cells are. You program a cell with nucleic acid, with chromosomes, and it begins to turn out proteins and do things. And so a virus is a little piece of nucleic acid getting into your cell and essentially replicating itself. And the diversity of mechanisms and the range of differences in viruses is pretty amazing and remarkable. It's one of these things of like every possible twist and combination you can imagine will have viruses do it. Particularly in terms of being DNA viruses, RNA viruses, the way they're getting into cells. There's a lot of viruses. And there are so many viruses out there. We were watching a video about, there's a doctor who uses a lot of big-ass words, and I don't. And he was talking about the virome and making some sort of comments about there being, literally quadrillion viruses in a spoonful of soil. I'm like, yeah, but these are bacteria viruses that are known as bacteriophage, and they have nothing to do with viruses that affect vertebrates, mammals. And so, it's easy to go kind of crazy on this. All right, so when we think about the viruses, though, they're not really living because they can't replicate in the absence of other cells. They are just bits of information. They have to have, there are components to the system that make it work. You have to have some kind of nucleic acid, and that's what we call the core. And this is where I say the possibilities are broad. You can have a DNA molecule, you can have an RNA molecule, it can be a single-stranded DNA or single-stranded RNA, it can be double-stranded. You can have a single RNA or DNA molecule, or actually several fragments of the RNA or DNA molecule. So pretty much there's a lot of different ways you can play this. We'll see it even is more elaborate than that. And this is what the core is. And then all viruses are going to have some kind of protective protein and membrane layer around them, which is the capsid. And so then we have, sort of the whole thing in toto is the nucleocapsid. I often think of my analogy with viruses as a corn dog, where we can think of the hot dog as the nucleic acid, and then the cornbread as the capsid and envelope. And then, depending, you can have proteins surrounding the capsid, and then you can even have things like the spike proteins that we see in the coronavirus. Another thing you have to have is some way for that virus to bind to the cell, and that is a receptor. And this is where viral specificity comes in, because if you ain't got the receptor, you can be exposed to the virus, and it really isn't going to do anything to you. It can't do anything to you. So that's why there are certain viruses in various animals that we care nothing about. I mean, the fact that you have a ton of viruses in the soil, bully for you, those are bacteria viruses. They don't bind to proteins on my surface. They can't get in and do anything to me, and even if they did, they don't have the right kind of programming to operate with me. So at the same time, as we are living right now, there are viruses that can cross species, viruses that have sufficient binding affinity for a receptor in a bat and a receptor in a human, because after all, mammals are more alike than they are different, and so it's not, in any way something that we shouldn't expect that you can have cross-species viral transmission. But at the same time, there are a lot of viruses that are very specific for very specific animals. So that's the receptor thing to be thinking about. And then we refer to them as being obligate intracellular parasites. They have to have the host to be able to actually carry out their replication. Just as a point, viruses don't carry or have the necessary machinery, if you think of it this way, for translating a RNA into a protein. They don't have ribosomes. They don't have tRNAs. They don't even have the ability to generate their own ATP. So it is simply a bit of information that you're inserting into the cell. They are filterable, but they are way smaller than cells. So, you have to have a very specialized kind of membrane filter, and it pulls a lot of vacuum, and it's going to be slow, but you can actually remove them. And some viruses are bigger than others. Are they living is one of the questions we can throw in. By the definitions of what life is, they probably don't fit every single bill, and so now they are information and not life. They are very diverse. And viruses have probably come about and evolved in multiple different spots and times. So here are just some examples. And one of the things that we've got these various sort of generalized terms for viruses, because viruses are so dramatically different. There's a range of different kinds of shapes, and trying to identify individual species of virus is, again, a bit of a trick, because the notion of a species, and it's a little bit the same with bacteria, the notion of a species is, can they genetically reproduce through some kind of sexual reproduction is one definition of a species, a pretty good one. And these guys aren't operating that way. And so we are now talking about species based on, whether they can infect the same cell type or how different is the genomic sequence. And so we have variants, and then I guess variants, as they begin to change long enough, become different species. But there are a range of different types of viruses that we can absolutely point out. So here's tobacco mosaic virus, which is a helical virus. These little rods, and this, as the name implies, actually infects tobacco plants. Plants have viruses. Pretty much all organisms have viruses. The poliovirus is an icosahedral virus. It doesn't have a lot of other stuff around it. The herpes virus is an enveloped virus, so it's actually not as cool a little structure as these other two. It's more of a blob. Let's see, Marburg, this is like Ebola, and it's got this weird little pea or a hook shape to it. And smallpox virus has got a fairly elaborate structure as well. One thing is that there are viruses that are found in a range of different cells, and just think about the icosahedral viruses, that we used to run a fruit fly cell line. And when you would hit it with molting hormone, that would cause the cells to go into mitotic arrest. And those kinds of things, like stress, cause the virus to begin replicating itself and packaging itself up in order to start moving out. It's like, okay, this cell that we're living in is reaching the end of its life. Let's go ahead and package ourselves up, rupture the cell, and begin infecting. And when in a electron micrograph cross-section of the nucleus, there were so many viral particles being produced that they would produce this absolute stacked and packed kind of structure to it. It's really amazing to see. So one cell can produce a whole bunch of viruses. So we've got a big range of sizes. So here's the virus that causes measles, the variola virus. Pretty big guy, and it's got a lot of stuff around it. So this is the nucleic acid embedded in this protein capsid, and then we've got a range of other things surrounding it, and then proteins on the cell surface. And as I'm looking at this, I'm thinking, how big is an average bacterial cell? And so this is 100 nanometers, or 0.1 micron, and the average cell is going to be around 3 to 10 microns. And so you really can't see viruses without an electron microscope. You might be able to, you can see it. You can actually pellet them in a tube and get kind of a white smear, but you can't physically see the way you can with bacteria. So just different viral structures. Herpes, rabies, got a slightly different structure. Variola must be mumps. Measles virus, the virus that causes AIDS, and here's one we've been looking at. SARS virus, this is the classic coronavirus. Ebola has got a pretty big package to it. And then we have things that are pretty small. Hepatitis, parvovirus, found in dogs. It's like causes kennel cough. So a whole range of sizes going on here, and different structures as well. So yes, you can filter viruses. It's not easy to do. You end up missing some, but you are able to actually identify and clump viruses up. So the bacteria, they're easy to filter out. Viruses, you're going to probably change the concentration a bit. So you have this solution now that is missing all the big stuff, but there's still a fair amount of viruses in here. So here is a norovirus that was identified on a cruise ship responsible for food poisoning. And you can see there's the pore, and then there's the virus. And so it's not all that easy to filter them out. All right. Now, a really good way that viruses can be seen is if they actually cause a lysis of a cell. And some of the very initial viral biology and viral models comes from looking at viruses that infect bacteria. And I hope in lab we actually play around with this a little bit, because I really love playing with bacteriophage. So this is a class of viruses that infect prokaryotes, and they're known as bacteriophage or phage. And the way they are able to manifest is that you'll have a virus bind to a bacterial cell, and things like the various carbohydrate receptors, the maltose receptor is a standard receptor for bacteriophage lambda. They will inject the DNA into the cell. The protein coat stays on the outside, and the viral DNA now can begin replicating. And we've got these two different terms that would write up, but I'm not going to, that we talk about, in that the virus can have a couple of different pathways. It can undergo what's known as the lytic pathway. In that case, this is where the virus is going to start replicating enough, and then fairly quickly blow the cell apart. And the lytic pathway results in basically a hole that this bacteria plate did look like a uniform plate about 4 hours ago. But then as the bacteria start to slow up, the phage say it's time for us to head out of here, and the phage had been replicating, and the bacteria that was carrying the phage was replicating, and so you had this little dot, this what is referred to as a plaque. And that's how you can see and count bacterioviruses, is by looking at plaques, and plaque morphology actually becomes a way you can identify different bacteria, or different bacteriophage. Now, the vaccinia virus, this is actually growing on a different kind of cell, on an animal cell. And you have, again, these places where the virus has lysed the cell. Not all viruses are going to manifest themselves this way, and we actually look at some interesting viral life cycles. But plaque morphology and looking at bacteriophage have been a fundamental tool and become sort of a model for how we think about viruses. And you can find bacteriophage, anywhere you find lots of bacteria, the soil will be filled with them. A great place to look for bacteriophage is sewage. So you take incoming sewage and you plate it onto bacteria. You take the sewage, and the virus is quite small, and so it doesn't spin down very easily. So you spin it down, and you have, and you remove all the, God knows what it is, material you can spin out. The solution will have a whole range of viruses. And you then begin taking and incubating a little bit of that fluid with a bacterial shot, culture bacteria, then plate it out on a plate, and you look to see if you're getting colony, or if you're getting plaques. And so you can identify bacteriophage. And there are so many bacteriophage that guaranteed, if you go looking at a little bit of sewage, you will find a bacteriophage that's never been studied before. So it is a very rich area for looking at new forms of genetic material, life, I don't know. All right, so we've got some classifications. And this is where we get into some of the different kinds of ways of thinking about viruses and how we can begin to organize sort of the classifications. We've got double-stranded DNA viruses. So this is DNA that would look like a standard DNA molecule, a plasmid, or a chromosome, double-stranded. We have DNA viruses, but they're single-stranded. Then we've got viruses that are RNA viruses. And these are double-stranded RNA viruses. We then have RNA viruses that are single-stranded. And just as a little note, we list plus and minus. If you remember, when we're talking about the genetics a little bit, we've got DNA is double-stranded. And the RNA polymerase is making a molecule of RNA off that DNA template. The DNA then can be thought of, the top strand is what the RNA is going to be looking like. It is going to be essentially an identical copy of the RNA. The bottom strand is the complement. And so RNA polymerase is actually reading the bottom strand to create a new copy of the top strand. The top strand is referred to as the plus strand. The bottom strand is referred to as the minus strand. The top strand is referred to as the minus strand. The bottom strand is referred to as the plus strand. It has to do with the fact that the RNA, when it comes in, if it is a plus-stranded RNA, that's an RNA that can actually be translated immediately. If it's a minus strand, it's got to be monkeyed around with. We'll get into it in a bit. All right, so single-stranded RNA viruses. The coronavirus that we know so well is a single-strand RNA virus. Has a lot, it is similar then in that way to West Nile, polio, hepatitis A and C, a variety of different guys. Then we have minus strand RNA viruses, and things like Ebola. The flu virus is one of these. Then we've got classes of viruses that are known as retroviruses, and they have a different kind of life cycle in that they actually are generating a molecule of DNA off of the RNA, which, if you think about our molecular dogma, that's backwards. We talk about the molecular dogma is DNA is transcribed to RNA. In this case, they're taking RNA and reverse transcribing it into DNA. So retroviruses are their own special kind of interesting class of viruses. HIV, feline leukemia virus, and then there are even double-stranded DNA RT viruses, which are just really weird, and Hep B is one of those. So we'll look at how these mechanics kind of played out here in just a moment as we start going into it. Another way you can classify them, of course, is how the way we were looking at the bacteria diseases is how they get in. And so we got a couple of examples that we can think of here pretty easily. We got food and waterborne. These are primarily getting in through the mouth, the nose, then making their way into the GI tract. And fecal contamination is a good way of getting these. This is why the good sanitation doesn't inhibit and block bacterial infections, but it also can affect the transmission of viral infections. Airborne is primarily now mouth, nose, and then getting into the respiratory system. So lungs versus swallowing it. This is probably going to be, inhaling air contaminated by someone sneezing or coughing. It's why we're wearing masks. What we're doing is reducing the amount of viral particle that we might be shooting out. And to a degree, you're probably reducing the amount of viral particle that you're inhaling. So then we've got things like STDs that are actually in the urogenital system. They're usually very delicate just because of the way they're being transmitted and the different kinds of places they're being taken up. A couple of different kinds of things that we have bloodborne. There are viruses that are transmitted by mosquitoes and things. And these are an interesting kind of game because you've got to get actually into under the skin, so a subcutaneous kind of injection. So blood-to-blood contact is something that you would have to be thoughtful about. And just as I'm thinking about that, what are some of the ways you can do that? I don't even want to deal with that. Micro skin tears is a way that happens. Sharing needles or being bit by a mosquito. So this is a fairly delicate kind of virus because they never are going to be exposed into the outside environment. So fairly delicate guys. All right, so we then have the basic component that we've been talking about. The core, which has the nucleic acid. Then we've got the protein capsid. There are things like the spike protein that are going to act as the interaction with the receptor of the cell. And then we've got the envelope. And so this term receptor, you're going to have some kind of binding interaction going on here. So you have to have a molecule on the cell that you're infecting, and then you have to have some kind of docking protein thing on the virus. So that's how we start thinking about it. And then some of the viruses, as we'll see here in a moment, can be enveloped in a plasma membrane. That depends on how they are making their way in and out of the cell. All right, so just to throw it in. Icosahedral, helical, binary, and complex, or these are the basic terminology for the types we talk about. And so, a naked virus, it's not naked in the sense that it's just a molecule of RNA or DNA. It doesn't have a plasma membrane around it. And so the naked virus, like an adenovirus, which can cause the common cold, you have the nucleic acid inside, and then you've got this set of proteins surrounding it that is the capsid. And then you've got these various spike proteins that are going to be interacting with parts of the cell you're going to infect. So when you start talking about viral infectivity and how infectious a virus might be, very often they're talking about structure of the spike protein. And so as variants of the coronavirus come around, the more infectious types tend to have slight changes in the spike protein. So this is a virus that is going to be released from a cell by lysing the cell. And so it doesn't have to make its way through the membrane. Now, if you're a virus that is making your way by not killing the cell, by getting out into the membrane, very often you end up being encapsulated in the cell's membrane. And so this is what is known as an enveloped protein. And it actually has a bit of membrane around it. So inside, we still have the genome, we have the capsid, and then we got layers of protein and other now glycoprotein and membrane components that make up the envelope. And so this is just a different kind of virus, and in part it reflects some of its different aspects of its life cycle. So with the filamentous guys, tobacco mosaic virus has got a series of proteins. It's a pretty simple virus. And then an RNA coiled up inside it. The flu virus is enveloped, meaning that it's going to have a plasma membrane around it. And there are a couple of different kinds of things that are known that are specifically viruses. There is different kinds of proteins that are floating around outside that are going to be interacting with different parts of the cell that it's infecting. And there is the RNA as a nuclear protein complex. And so this is sort of a cartoon of it in a little bit simpler form. The nucleic acid and the lipid bilayer. So this is a envelope type protein. They can have a variety of different kinds of binding things like hemagglutinin and neuraminidase, which are responsible for binding and then actually allowing it to burrow into a cell. So viral shapes continued. Lots of different viral shapes. So tobacco mosaic virus as a filament. The icosahedral types that are naked, non-enveloped, and enveloped have the membranes, but still have a range of glycoproteins on the cell surface. And then some of my favorite, the complex guys. This is a classic bacteriophage. It looks a little bit like a lunar lander spaceship. And the DNA is held in the head, and then you have this tail, and then these sort of little fibers, little feeders that make contact with the cell. And this is how the cell receptor actually works. And so you can get, electron micrographs of this kind of stuff. It's not really easy to do, but it's not that hard to actually generate an electron microscope image of it. All right, so we got a range of different shapes that fall out from the different kinds of life cycles. And I'm thinking about going to the board, but not. All right, so this is some of the definitions of viral life cycle that has been come about from bacteriophage. And so as I mentioned, the bacteria viruses as a model system is where we've learned a lot about the way viruses work. And so there's a whole range of terms that come from bacteriophage biology. And so the first step is the virus actually has to bind to the cell, this adsorption notion. And you do this by taking a cell culture and shooting in 100 microliters of this sewage fluid and letting it sit for a little while, 10 or 15 minutes. That allows the the viral particles to actually bind to the cell. Once that has happened, the viral chromosome is able to actually penetrate into the cell, and we get into the cell, and this is now where the viral nucleic acid starts to get transcribed and translated, begins to produce its own proteins. And there's a whole bunch of really cool stuff that we really won't get into in terms of the control, because the virus has a couple of different ways it can operate. It can simply, in this case, just begin replicating extrachromosomally. You get a bunch of viral chromosomes generated, and then they begin to generate an RNA. That RNA is translated, and you start to produce all the protein code. And so I think it's a little bit like you have this information that comes into the cell that is able to dictate the construction of its own suitcase. The suitcase is built, it's opened up, and the RNA, or the DNA, jumps in, closes the lid, and now that suitcase can be shipped back out. And it is an auto-generating system. And so the proteins that it produces are referred to as packaging. And it is an autocatalytic process. It will just begin to self-assemble, which is kind of remarkable, but that's what it does. So the viral chromosomes are surrounded by their coats, and then you have lysis. And so in this case, this is going to be a non-enveloped type system because you are literally popping open the cell, and hundreds of viral particles are moving out to infect the neighboring bacteria. And so that's the way this life cycle works in what's known as the lytic phase. So a lytic bacteriophage is really a fairly simple kind of system in that it infects, replicates, makes more of itself, assembles, and then goes back out. This is in comparison to another kind of life cycle that we're going to call a lysogenic life cycle. So this is just a more of a diagram, sort of an interesting, cartoony looking guy. We've got, in bacteria, a lytic life cycle in which the bacteriophage lands, injects the DNA into the cell, and the bacterial or the phage chromosome is replicated, phage proteins are being generated. The phage will physically pop open and kill the cell. Thanks a lot. After all I've done for you, you're going to lyse and kill me, and the phage now can replicate, or it can spread. And this is why you end up with a plaque, because you actually kill and lyse that bacteria. In a eukaryotic system, it's a little bit more complicated, but still we've got the virus attaches. In this case, it's brought into the cell through endocytosis, so it's not injecting its DNA or nucleic acid directly. It's actually being enveloped and brought in. It then is going to be releasing its viral RNA. The RNA is able to be translated into protein, and the proteins is now start to build all the components to the virus, and the virus can begin to package itself up, and then it is released. So, a different kind of system. The other side of this is something that's called lysogeny. So, it's a way of just, the two terms are you have the lytic cycle or the lysogenic cycle. And lysogeny is an interesting game in which, rather than lysing the cell and generating viral particles immediately that will lyse the cell, the virus actually integrates into the host genome. So the lytic cycle is just really fast, producing lots of virus, they rupture the cell, and you go. The lysogenic cycle is a little bit more subtle, kind of a sleeper agent here, in that the DNA inserts itself into the bacterial chromosome. And now the bacteria just divide. They don't know that they have been infected by this virus, and they are dividing, and the offspring also now have the virus incorporated into its chromosome, and it is simply a ticking time bomb waiting, because the phage are going to say after a while, oh, growth is limiting, the amount of food is starting to limit, you're starting to slow down, I don't like you, and they will then excise themselves from the genome, package themselves up, and go into the lytic cycle. And so it's one of the things with viruses is that, the virus might, sort of say to you, listen, I'm going to be cool. I'm going to become a lysogen, and you won't even know I'm here. I'm just going to replicate along with your chromosome. Don't you worry about me. I'll be quiet. But viruses, you can't trust a virus. They will eventually become lytic. And lysogens actually happen with the measles. And so this is actually what causes shingles. And so if you've had the measles, you have measles lysogens in parts of your neurons. And they're saying, oh, I'm not going to bother you. You won't even know I'm here. I'm just going to replicate along with your chromosome. Don't bother me. Don't bother about me. But under stress, they're going to go, oh, things are kind of looking a little rough right here. Let's go ahead and package up some virus. Go ahead and get out of here. Oh, great. And you have, a herpes outbreak. You have shingles. So the bottom line is never trust a virus. They say they're going to be nice, but it's like a scorpion. Man, they eventually are going to bite you. So herpes is a classic latent lysogenic virus. It actually infects neurons, and it can sequester itself in the neuron. And so you've got essentially this latent period in which the virus has incorporated its genome into the existing cell, and so it's a little sleeper agent. And the immune system isn't going to attack it because there's nothing happening there. It's only when you have this acute phase of the virus actually replicating and now packaging itself up and escaping that the host immune system can get a hold of it, because now it begins to see it. So this is how you get recurring cold sores, is that you never can clear your system of the virus. It's always there as, don't mind me, I'm just a little lysogen here. So another thing to think about is that viruses, the way they replicate, they don't divide. They are built. They're assembled. And so when we look at bacteriophage particularly, the number of infectious particles, it rises pretty steeply. And you then have a period of where the virus is maintaining itself as a lysogen, and then there's going to come a point where it is going to go into the lytic cycle. And that's where we start to see the phage secreted, they ruptured the cell. And so we have the initial accumulation, but then over time we start to see bacteria lysing. And just as a term, we have the lytic cycle, which is the bad news. You're going to pop them open. And then we have lysogens, which is when they incorporate themselves into the cell and the genome. So that's something to keep in mind of the lytic versus the lysogenic cycle. And I'm going to stop here because I feel like it. So we'll get into some antiviral drugs and viral therapies. Interesting to know that we ain't got a lot of antiviral drugs, so vaccinations, man, that's probably the best answer for dealing with viruses. That and maintaining a good immune system. All right. Ladies and germs, I'll catch up with you later. Ciao.